Good evening, my name is Jason, and today I'll be talking about the cancer drug Taxol, known by the generic name Taxol. Taxol is an anti-tumor drug that is based off a plant alkaloid derived from the Pacific yew, also known by the name Taxus brevifolia. Taxol reduces the critical concentration of purified tubulin subunits necessary for polymerization into microtubules in vitro and increases the percentage of tubulin subunits that assemble. Furthermore, microtubule polymerized in the presence of Taxol are protected from the disassembly normally induced by cold or calcium treatment. The effects were in stark contrast to previously identified microtubule poisons, including colchicine and vinca alkaloids, which prevent microtubule polymerization. Similar effects to its effect on purified tubulin. Similar to its effects on purified tubulin, Taxol promotes microtubule polymerization and stabilization in living cells, where it is capable of antagonizing the effects of colchicine and vinca alkaloids. Phenotypically, Taxol treatment arrests a diverse array of cell types in mitosis in both animal tumor models and cell cultures. A large majority of reports indicate that Taxol arrested cells are in metaphase and contain near normal bipolar spindles. Images of Taxol treatment show cells with chromosomes aligned at the cell equator, although in some cases one or a few chromosomes have been reported to remain misaligned. A small number of studies have reported that certain concentrations of Taxol induce multipolar spindles. However, until recently, there was no evidence to suggest that these effects were clinically relevant. The dominant perception by far has been that the anti-tumor effects of Taxol occur due to the ability to arrest cells in metaphase or bipolar spindles. Taxol-induced mitotic arrest occurs due to activation of mitotic checkpoints, also known as the spindle assembly checkpoint, the major cell cycle control mechanism acting during mitosis to prevent chromosome missegregation. The mitotic checkpoint delays separation of the chromosomes, which enter mitosis as replicated pairs of sister chromatids, until each pair has made stable attachments to both poles of the mitotic spindle. This arrangement ensures that each daughter cell will receive one copy of every chromatid. Chromatids connect to the spindle microtubules through their kinetochores, protein complexes that assemble on centromeric regions of DNA. Unattached kinetochores, which have not made stable attachments to microtubules, activate a signal transduction cascade that delays mitotic progression by inhibiting the anaphase promoting complex cyclosome. Taxol treatment arrests cells in mitosis due to the presence of a small number of unattached kinetochores. In addition to its ability or utility in cancer therapy, Taxol is also widely used in cell biology. In untreated cells, bipolar attachment of sister chromatids places kinetochores under tension, which helps stabilize the interaction between kinetochores and spindle microtubules. Taxol treatment reduces the tension in kinetochores that maintain bipolar attachment and is a useful tool both for arresting cells in mitosis and for dissecting the contributions of tension versus attachment in satisfying the mitotic checkpoint. The side effects of Taxol include temporary hair loss, flushing, nausea, drowsiness, joint and muscle pain, and injection site reaction. Seeing as it's a anti-tumor drug which is supposed to arrest growth of cancer cells, it is also expected that these drugs or this drug Taxol would affect other cells that divide rapidly in the human body, such as cells that line the human stomach, and hair cells that actively divide at rates that are similar to those of active cancer cells or active tumor cells. One of the questions I did have upon doing this was if there were other drugs that had similar effects or modes of actions to Taxol, but didn't have the exact cytotoxic effect on regular cells, like say cells of the stomach lining or hair cells. I was also curious if there were other plants or fungal alkaloids being researched with anti-tumor abilities. In the spring semester, I was talking to Dr. Pedroza, and I know he and Dr. Lyons were working on finding fungal factors that affect tumor growth, so that might be something that may help answer the question.